This, I'm recording so everybody's understand. Sit next to me so you know, so you see what I'm looking at. You're gonna start off on. Oops. Someone close the door. Nope. Doesn't matter. Page 17, and you're gonna read about enzymes. Let me redo this. Page 17, you're going to read, start off with enzymes. You're going to look. We're going to do a lab next week after the second half of our final, or you're going to do a lab on catalase. So you're not going to be spending the full 2 hours on a multiple choice exam. You're actually going to do a lab, and I'll be grading you on whether you're following safety procedures, which we'll go over probably Friday, and whether you're using your scientific method appropriately. So lab catalase. In the introduction to catalase, you're also going to read today. Catalase is an enzyme. You're going to pay extra attention to page 19. That's page, by the way, that was page 17 and 18. So for the lab, catalase lab, you're going to look at page 18. Wow. I know. I don't know what to say. And you're going to label what you're going to do in the book, in the workbook. You're going to label everything for number one. So you're labeling. And they have a list. They have a list of catalase, active site, hydrogen peroxide, substrate, products being released, oxygen gas, and water. You're gonna label everything. Then you're gonna go ahead and annotate the graph. For 2, you're gonna annotate or label the graph. You're gonna label the reaction with the enzyme, which is the curve. You're gonna label without the enzyme. You're gonna look at activation energy. So in order to be able to label all these things, you have to actually read. Then on page 21, is the lab that you're going to be doing next week during your final. All right, there's going to be a whole setup there. You'll be using that in your setup. And there's a data table. You're not going to have to worry about writing your own lab write-up. You're just going to fill in the data table, answer the questions. So you're going to have to do that. So for homework over the weekend, you're going to look at page 21 and make sure you're ready to answer all the questions by what is it you don't understand? What is it you do understand? Does everybody understand that? Yes or no? Yeah. Okay. So that's 21 through 24. On page 25 is your experimental design. And now you're what you're gonna do in the experimental design is you're gonna look and read after you've done all your reading, after you've looked at pages 21 through 24. You understand what's going on with the lab. You're gonna start thinking about what are some variables you might want to mention. Follow the teacher's procedures determining research question you want to investigate. You're gonna have to actually design an experiment. That's part of your final. You're doing this mostly at home and in class. I'm here tonight. Tomorrow night, if everything goes well, we'll have a chess tournament, so I won't be here tomorrow. But we'll have Friday as well in class. I know it's short, but I think we'll have a regular class period. So ask me questions, and you'll see that you're designing your experiment all the way through page 34. Okay? Is that clear? So 25 through 34 is basically half of your final that you're taking home and working on it. You're working on it in groups. You're asking me questions. You're going to a tutor. There's tutors downstairs. All next week after school, there's tutors here. This part of the final you can even turn in by Thursday of next week if you need more time. Okay, so you can see me every day after school next week if you want. Am I clear on this? So this part of your final is an experimental design. You'll have time in class to actually do the lab on page 21 through 24. You're going to actually be doing the lab. But the experimental design part, you're going to design it on your own or in groups. You can have the same answers as someone else as long as you put down that you're working with them, but everybody has something to turn in. Is that clear? Does that make sense? Yes? All right. We're all good with this so far? Scroll up. Is that making sense to everybody so far? So this is going to have a due date latest possible. Honestly, you could probably get this done today, but latest possible Thursday of finals week. If you can't see, move, please. Is that good? All right. So again, half your final is multiple choice, fill in. It'll be on computer. The other half is going to be a lab and experimental design. Now, for today, what is it you have to get done for today? Give me a second. 30, that's pages 17 through 18. I'm going to highlight everything that's due today. Pages 17. I like yellow highlighter. Pages 17 through 18, read enzymes, intro to catalase, right? And label the graph and label the things that they tell you to label on number 1 and 2 on page 18. This is today. All right. This is due on the latest Thursday of finals week. It's experimental design section. It's on pages 21 through 24. There's a lot of that. 21 through 24 is the lab. This will be done on finals week. You know what? let me make it clear by cutting this out. So this is going to be on the day of your final. It seems complicated, but I think it's going to reduce your stress a little bit if you're actually doing something rather than sitting still and doing multiple choice for 2 hours. Does that make sense? And the rubric I'll show you in a minute on grading the lab on experiment day, you're not going to be graded on whether it works or not. That may or may not work. What you're going to be graded on is, do you know what you're doing? Or do you have to have someone tell you? Have you read the lab or not? Like, that's number one. Do you break any safety rules? If you break any safety rules, you failed that portion of the final. Am I clear on that? Okay. And we'll go over safety on Friday. It should take no more than half an hour, and I'll give you a safety contract. No safety contract, you can't do the lab. For today, another thing you have to have done for today, and I'll print this out for you too. I'll have a nice little flyer for you as always that'll have everything listed for you. But now you have it in front of you. Then also for today, after you've read your Catalay stuff, after you've read this, so this is part 1 for today. Part 2 for today is to do pages 35, 36, and 37. That'll show me if you understand what a catalase is and what an enzyme is and how it works, okay? Now, pages 39 through 72 is classwork and homework due on the day of the final. For you, that's Monday. These are practice questions, final practice questions. Is that clear? So page 39 through 72 are all practice questions. Don't worry, it's not that much. It looks like it's a lot, but don't worry about it. You should be able to do everything. Any labs, if they tell you to do a lab, obviously you're not doing a lab. Unless you feel like doing it, you want to come in after school and do it, you can't with me. Okay? Are we clear on what we're doing? I don't need to. So you're not going to have me lecturing to you today. Today's activity is broken down into 2 sections. Part 1, you're reading. What are you reading today? 17 through 18. You're labeling. If you need help, you're going to come and see me, or you raise your hand. On the finals day, you know what the final is. Today, part 2 is to finish 35 to 37. Obviously, it's due today by the end of the period, but you can turn in by Thursday morning if you need to, right? The only things that I'm going to grade next week is pages 39 through 72, and then your final. Okay? Yes? All right. All right. So there you have it. You can work in groups. I'll stop talking now. So because people want me to explain what an enzyme is again, I'm going to quickly do it here, and then you guys, it should help you as you're doing your reading. So an enzyme is usually a protein. Is usually a protein. It can be RNA. It makes reactions go faster. And the third thing about an enzyme is it's not used up in a reaction. So now for an example, when I'm just going to go ahead and talk about the example they're giving you in the book. Hydrogen peroxide, everybody knows what that is. H2O2. That's the chemical formula for hydrogen peroxide. Very dangerous stuff. It kills everything. Nothing survives this stuff. It's very reactive. It'll bleach your clothes, so be careful. H2O2 is so deadly that almost every organism has an enzyme to break it apart. No living thing wants H2O2 around it. So there's an enzyme, and I'm just going to draw it like this, like a Pac-Man, that's going to break this up. And it's going to break it up into oxygen gas, and I made the mistake of thinking it was hydrogen gas, oxygen gas, and water. Now when I balance the equation, just put a 2 here. That makes the hydrogen on this side equal 4, the oxygen on this side equal 4. The oxygen on this side, if I put a 2 here, that's the hydrogen's a 4. Do you agree? Because 2 times 2 is 4. 2 times 1 is 2, plus 2 is 4. So at 4 and 4, the equation is balanced. Everybody see that? The same number of elements on both sides. Are we good? All right, so this is the reaction. You're going to take this hydrogen peroxide with the enzyme, and it makes oxygen and water. This is what happens when you pour hydrogen peroxide on your skin. The cells in your body have catalase. This enzyme is called catalase. And it breaks, its only job is to get rid of H2O2 because it's so dangerous. And every organism has it. So what does this enzyme do? By itself, if you look at a bottle of hydrogen peroxide, how long do you have before it goes bad? Hydrogen peroxide. You all have hydrogen peroxide at home? How long do you replace it every week? It lasts for a long time. It doesn't break down by itself. It doesn't turn into water and oxygen by itself. It does a little bit. You'll see little bubbles. And that's how we're going to tell if something's happening, because we're going to see what? If you have a liquid and it starts to turn into oxygen gas, what are you going to see? Bubbles. Each of the bubbles is oxygen gas. The rest of this is, some of it is H2O2 and some of it is H2O. But they're mixed up together, so it looks like one thing. It's a solution, and you get some bubbles, but not a lot, very few. So the reaction is very slow. So if you took a graph and you put in the amount of energy that you're that requires for the reaction to go on, is going up that way. And here's how the time it takes for the reaction to go on. What you're going to get is, you're going to start off with H2O2. It's going to require some kind of energy. And then after you get some amount of energy going, it will become H2O and oxygen gas. Now, this amount of energy required is called activation energy. This is called activation energy. Do you agree that this paper will burn? Do you agree this paper will burn? Everybody? Everybody agrees that paper burns? How long am I going to wait for it before it burns by itself? If I just sit here and watch it, how long before it starts to burn? A long time, right? But what happens if I take a lighter and I add a little bit of energy right over in this corner? The reaction happens, right? That's called, you can think of it as a spark. Almost every reaction needs a spark, a little bit of energy. When you turn on your car, what do you hear? You turn the key. What happens? Energy. You have the battery in your car shoots a little bit of electricity to the engine, and it lights up the gas. You go, vroom, right? That kicking is all the electricity that's being shot at the gasoline. When you turn on your stove, you hear click, 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 then whoosh, right? If you have a gas stove. Anybody here not ever hear that on a gas stove? When you hear click, 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 if you look, you'll see a little thing you actually have a battery in the stove that's shooting little electric sparks. This spark is necessary for almost every reaction. It's called activation energy in order to get that thing over the hump. Once it's up here, though, it's no problem. It'll take care of itself. It'll run. Once this starts burning, I don't have to add any more. I can put the lighter away. It'll burn all by itself. Same thing with hydrogen peroxide. Well, what the enzyme's going to do is it's going to lower that amount of energy. So when you have it in a bottle, if you leave it alone, this, as you know, everything has energy, right? Everything in this by itself, without adding any energy, this beaker has kinetic energy, correct? But it doesn't have enough to make H2O2 turn into water and oxygen. So what could we do? We could add energy, right? In the form of what? What if I took hydrogen peroxide and I added heat? What would happen? It would turn into what? Water and oxygen. I can make it turn to water and oxygen, correct? Very easily by adding heat. That's not something I want to do. Because if you drink hydrogen peroxide, in order for me to make that not poisonous and make it into water and oxygen, I would have to put you in a fire. Would that save your life? No, you'd burn. That's no good. So you have an enzyme called catalase that lowers the activation energy. So at this same temperature, it turns into water and oxygen. Is that clear? And the enzyme is a protein. It has a specific shape. And you have a crazy diagram, a crazy picture of what are called domains. So proteins are all folded. They're folded in a specific shape. I've been giving you really simple shapes like the Pac-Man shape I just showed you. But your book gives you what's called the ribbon drawing. So each of these domains has a different set of amino acids. Sometimes there's different polypeptides, but it makes a shape. That's more like what the enzyme looks like. That drawing is actually a drawing of the enzyme, catalase. So there it is. That's all the intro you need to get this done. Remember, an enzyme is just lowering the activation energy. As you're reading, that's all it's doing. It's usually a protein. By the way, because it's part of your review for your test, you should know that an example of an enzyme that is RNA enzyme, so not a protein, but RNA, is an organelle known as a ribosome. So a ribosome is actually an enzyme. And the reaction that it speeds up is amino acids Remember, there's a bunch of amino acids in a long chain, taking amino acids and making them into a protein, a polypeptide. So you're linking the amino acids together. That's what ribosomes speed up. That's the end. So that's a review for the test, because you should know a ribosome makes polypeptides. We'll go over when we get back in January how that happens, because you will be tested on that on your OST. But that's a story for another day. For this test, all you have to know is a ribosome makes proteins, makes polypeptides. That's it. They're found on the rough endoplasmic reticulum, RER, and in the cytoplasm. Okay? Are we all good with this? So there it is. Go ahead. Good luck reading.